So good morning, students. Previously, we are completing this structures of the reproduction. Na? I mean, human male and female reproductive systems, cell division processes, okay, and flowering plants, arrangement, process of fertilization, pollination, up to the formation of the seed also. This is a new topic also today. Cell division and continuation of life. How the cell division takes place for the continuation of life of an organisms, even in unicellular and also in multicellular. What is meant by cell division? How the cell division process is going on? What is the importance of cell division for the maintenance of the life of an organisms? Okay, clear? To know the complete information regarding this cell division, number of scientists did a comprehensive works on this particular one. Okay, first one, Rodolph Virchow, Robert Remak, Walter Fleming, Wilhelm Rux, Gregor Mendel, and August Weismann. You know, in the ninth class syllabus, cell division process, plant cells and animal cells. Plant cells were discovered by Schleiden, and animal cells were discovered by Schwann, Theodor Schwann. Both Schleiden and Schwann also proposed cell theory statements. Cell theory statements means all the cells are existing from their pre-existing cells. All the cells have the same components of the one. They have the same similar components and having the similar functions. These are some of the properties. These are some of the theories of the cell division now. Okay. But the third scientist after this particular Schleiden and Schwann, Rodolph Virchow. Okay. Rodolph Virchow first of all observed cell division process. Cell division. To realizing this, he also implemented a statement is called as omnis cellulae decellulae. Omnis cellulae decellulae, which means cells existing from pre-cell. All the cells arising from pre-existing cells. Cells arising from pre-existing cells. See this? So for example, this is the cell 1. This cell can divide into 2. And this is the cell again divides into 2. And this is the cell again divides into 2. These two cells are existing, are producing from the existing cell. And again, these two cells are producing from the pre-existing cell. See, the statement is clear, very clear now. Omnicellulae, decellulae. All the cells are forming from their pre-existing cells. The first statement regarding the cell division first given by Rudolf Virchow. Rudolf Virchow. Next, Robert Remak. He is opposed to the statement is called binary fusion. Not fusion. Binary fission. The first topic of the reproduction. Unicellular organisms, mostly Paramecia, amoeba, and euglena, and including the bacteria also, they divide their bodies, they divide into two. By splitting into two, by the process is called as binary fission. Here the word binary means two, fission means division. One organism is splitting into two parts, two organisms, two successive organisms. Clear? Such a process commonly called a binary fission. So, how the binary fission is going on? One cell is exactly, so all the cells are divided by the process of binary fission said by the Robert Remak, the German scientist now. So, the cells are also divided into two equal splitting part. Okay, at the same time, the two cells which were producing from the pre one having the same components of the parent one. Such a kind of a process of binary one, first of all observed by the Robert Remak. Okay. Next, Walter Fleming. Walter Fleming. He is opposed to the first statement is called as mitosis. Mitosis. You know, in the 9th class syllabus, mitosis and meiosis process. Mitosis and meiosis process. Here the mitosis process, one cell divides into two. Okay. Whereas in meiosis, one cell divides into 4 equal cells. But at the same time, comparing that chromosomal numbers automatically changes from the mitosis to meiosis. Clear it is. So first one, mitosis process first implemented by the Walter Fleming. According to his observation, the mitose means the fine threads. See this? Mitose means a fine threads. So what kind of thread-like structures are present in the cell? See that. So for example, this is the cell. During the process of cell division, this is the nucleus. Within the nucleus, a chromatin material is present. Chromosomal structures are present. These chromosomal structures along with chromatin materials, okay, also looking like a fine thread-like structures. Also fine thread-like structures. These fine-like thread-like structures are split into two equal sharing parts. Such a process are first of all said by mitosis, said by the Walter Fleming as a mitosis. A fine thread-like structures are divided into two equal sharing bits. These fine thread-like structures can also carry the genetical information, but you don't know completely what is this fine thread-like structures, how they are carrying this information. According to his observation, a fine thread-like structures can automatically separate it into two one. Okay, so this process was named as a mitosis. Later, these particular structures called as the chromosomes, called as a these bodies are commonly called as the chromosomes. Here, what is the meaning of the word chromosome means? Here, the word chromo means color, soma means body. So the colored bodies are present here within the nucleus. They are sharing into two equal parts. These colored bodies also plays a major role in the process of cell division. Was first said by the Walter Fleming. Such a process commonly called as a mitosis. Okay. Then Wilhelm Rex. These fine thread-like structures called as the chromosomes. He giving the clear importance regarding this particular chromosomes. So here the word chromo means colored, soma means bodies. Chromosomal structures are first of all clearly understood and given complete detailed information by Wilhelm Rex. See that first one. Within a cell, this is the nucleus. Within the nucleus, for example, a colored body. This is called as chromosome. A colored bodies are present. So within this particular colored bodies, a fine structures. This chromatin material is completely present here now. These fine thread-like structures are also called as a DNA. DNA. These chromosomes carry the genetical information by this kind of a structures called as a DNA. First of all, the Wilhelm Rex observed the chromosomal bodies. Chromosomes are not a single bodies. These are the called as the colored bodies, which also carries the complete genetical information segment called as a DNA. A small segment of the DNA is called as a genes. Later, these DNA structures and the genes were implemented and understood by the James D. Watson and Crick. Okay, so let's listen. These colored bodies can also carry the genetic information structures called as the DNA. Okay, so number of chromosomes are present is the number of chromosomal number may varies from the organism to organism. Organism to organism is particular species of the organisms having the particular number of chromosomes only. So one curious important point here, for example, this is the cell. The cell contains, for example, 20 chromosomes. In each and every mitosis process, these 20 chromosomes have to be doesn't change for in each and every successive divisions also. And again, once again it is divided by, and again 20 chromosomes, and again 20 chromosomes. Once again it is divided by the mitosis process, for example. And again these four cells also having the 20, 20, and again 20, 20 number of chromosomes only. The number of chromosomes should not be changed. This clarification is given by the complete August Weismann. Once again, listen. So these chromosomal bodies first of all observed by the Wilhelm Rex again. Next, George Gregor Mendel. Mendel first of all hypothesized genes carries these particular segments of the DNA called the genes are heritable characters. Inheritable characters. Do you know inheritance? Inheritance. Do you know what is called inheritance? Transferring the characters or the traits from one generation to another generation. Transferring the traits or characters from one generation to another generation. That is commonly called as inheritance. See that all the time. Mango plant gives a mango tree only. Of course, the mango plant once again. Coconut gives the coconut plant once again. Human being gives the human being only. Tiger gives the same kind of a species of an organisms. Tiger won't give any kind of a fox now, and again fox does not give any kind of an other animals because these kind of a segments of DNA, these are commonly called as the genes. The segment of DNA, for example, double helical structure called DNA is present in the human structure now, human cell structure. So a small segment of the DNA, in the small segment of the DNA, that's my segment of the DNA called as a gene. These genes can carry this particular traits. Okay, for example, that boy is looking same as it is like his father. That animal is same as it is looking like his parent one. How it is possible? These genes can carry these particular traits. The traits exactly copies to the coming offsprings. Okay, such an information of the genes is said by the Gregor Mendel. Such a characters which carrying from the parents to the offsprings or from the generation to generation, such a one is commonly called as inheritance. The inheritable characters are transported from generation to generation. Okay, so he also commonly called modern plant father of modern plant genetics. Okay, next one, August Weismann. August Weismann clearly said this: in each and every successive divisions, in each and every successive divisions, chromosomal number of an organism should not be changed. Chromosomal number of an organism should not be changed. Okay, same number of chromosomes should be maintained in each and every successive divisions. And again, all the organisms carries the same number of chromosomes to the coming generations. Okay, so for the clarification of this one now, so for example, human cells, take an example as a human cell, we have 23 pairs of chromosomes. 23 pairs. So for example, for our easy understanding, we have 46 number of chromosomes. So this number of chromosomes should not be changed in any successive divisions. Okay. So for example, if we are taken, this is called as a mitosis process. Before mitosis process, the number of chromosomes of the 46 is as a double. Okay. This one as a 92. 92 number of chromosomes. Okay. When the cell divide into two equal bits, and again the 92 as sharing as a 46 and again 46. The 46th number is called as the original number of chromosomes present in the parent cell. And now these two cells are ready to divide as a mitosis process once again now. So for example, these two cells are ready to divide once again. Before the division, the cell number of this one, of course the chromosome number again divides as a 96 and again this one as a 96. And again this can gives the two cells once again. This cell gives the two cells and this cell gives also two cells now. 96 shared as a 46 and 46. 46 and again 46. Same number of chromosomes should be maintained in each and every successive divisions. Okay, in every generation, the number of chromosomes should not be changed. Of course, the number of chromosomes might be changed. There might be a complete abnormalities. Number of abnormalities may takes place within an organism. Okay, we can't imagine what is the shape of an organism. What is the color, size, shape, or structures? We can't imagine. Entire world should be collapsed. All the organisms have to maintain to a particular number of chromosomes in each and every successive divisions for millions and millions and millions of years continuously again. You understand? So this is the process of mitosis. One more thing. So come to the point as a meiosis. A slight difference between the mitosis and meiosis. But in meiosis, the mitosis process occurs completely or occurs only in the somatic cells, except the reproductive cells. Whereas in the meiosis, for example, if you are taken example as a human cell. So for example, here 46 number of chromosomes. Okay. Before cell division, for example, this is the male gamete. For example, this is the male gamete. This is the male gamete. So just wait. So during the process of meiosis, this is the cell. Number of chromosomes are 46. Before cell division, basically during the meiosis, totally 4 haploid cells are producing. Basically during the process of mitosis, 2 diploid cells are producing. Number of chromosomes are same, diploid condition, but the number of chromosomes are reducing by half. With the process of meiosis, the number of chromosomes is reducing by half. But here, producing all the cells are haploid. So first of all, the number should be as 96. Okay. Here the number is 46 and again 46. And again this cell should be divided as a 2. Now 1, 2, 3, 4. And again the 46 number should not be doubled here. Once again. So here the number should not be doubled. Here as a 46 as a 23, 23, 23 and again 23. Now the entire process is called as one meiosis. Now this is called as one mitosis and again this is one mitosis and this is one mitosis. But entire step is called as one meiosis. So observe here the parental chromosomes has the 46. Before division that should be doubled. 46 and 46 again, for example, for your understanding. So and again after the final division 23, 23, 23, 23. These all cells have the haploid set of chromosomes. So for example, these are all called as a male gametes. For example, same number of cells are also present here. For example, this is the female gamete cell. These are male gamete cells. And again 46. For example, here 92 again 46, 46. And again this cell should be divided as a 2, 1, 2, 3, 4. For example, 23, 23, 23, 23. For example, these are all called as female gametes. Male gametes and female gametes. For example, 23 number of set of the chromosomes with male gamete, 23 set of chromosomes with female gametes. If these two are fused, do you know what is the process of fertilization? Fusion of male and female gametes combining together to form the original number of chromosomes. Here 23 set of male, here 23 set of female. When these two are combining with each other, okay, 23 pairs, or for your understanding, these number is commonly called as the 46. 46, this is the original number of chromosomes present in the parent cell. Clear it is? Now this can have the chance to formation of the zygote here. With only having the male gamete, with only having the female gamete, there is not possibility to form the zygote here. For the formation of zygote, single set of chromosomal number should from the male, single set of chromosomal number should be female, should be compulsory. When these two chromosomal numbers, single set of chromosomal number should be combined with each other, the zygote should be formed. The zygote having the original number of chromosomes should be present in the parental cell itself now. Now, this can also lead to formation as a zygote one. Okay. See that? So, for example, this is the zygote cell. The zygote cell can divide as a mitosis process once again. All the time. All the time. Zygote continuously have the division is called as a mitosis. Now, this is diploid set of chromosome. Diploid set of the cell. This is two cells. Two cells dividing as a into 2, 4, into 2, 8, into 2, 16, 32, 64 like this. This like uncountable divisions takes place. Some of the cells from the zygote forms as a mass of cells. Some of the cells forms as a mass of cells. Some of the still some more cells. For example, like this. All the cells forming from this. And again as a 2n. Diploid condition, 2n. Diploid condition, 2n. Diploid condition, 2n, 2n, 2n. A mass of cells from the zygote can forms as a mass of cells. For example, these mass of cells having the diploid condition forms as a bone. For example, these mass of cells can form as a, for example, muscle. These mass of cells can also form as a, for some more organ, just like as a digestive system, for example. These mass of cells can also form as a respiratory system, for example, or a heart, for example. So these mass of cells also can form. One more, some of the cells, for example, these cells, for example, these cells commonly called as immature cells. These immature cells can also form divided as a gametes. For example, as a male gamete, for example. Sometimes the immature cells can also form as a female gamete. One more thing, keep it in your mind. A kind of an organism, either it is, for example, male or female. If this organism is a male, this can forms from the immature cells as a male gamete. If this organism is a female, this can forms as a female gamete. For example, egg. This is called as a sperm cell. This is called as a egg cell. Only from the male, there is no possibility of formation of sperm and egg cells. Only from the female body, for example, there is not possibility to formation of sperm and egg cells. For example, if this organism is a male, they can form as a sperm cells. If this organism is a female, from the immature cells, they can form as the egg cells. Okay, egg cell having the haploid set of chromosomes N, sorry, sperm cell, and again egg cells also having the haploid set of chromosomes as N. When the combining with N and N, for example, these two combining as the 2N diploid condition, zygote is forming now. Now this diploid condition zygote can also form as a new organism and also same like this now. Some of the cells can also form as a bone. Some of the cells can also form as a muscle. Some of the cells also form as the digestive system or the heart or the lungs or the respiratory system, anyone. And again, from this one, some of the immature cells can also form as a gamete cells, either the sperm cells or maybe the egg cells itself now. Clear it is. This kind of the continuation of cells for every generation takes place. This point is said by clearly scientist is called as August Weismann. August Weismann. Okay. So to following this topic, what is called cell division? Cell division in human beings. How the cell division in human beings takes place? Okay. All the animal life is started from a single cell is called zygote. Diploid condition cell. Diploid condition cell called zygote. From the zygote, the human life, not only the human life, any animal life, mostly in the animal or plant kingdom, is started with a single cell called as the zygote from now. Okay, during the development of the zygote, automatically zygote to embryo, embryo to fetus, fetus to baby, baby to automatically, and the further animal is not forming now. Okay, so then, for example, human being is the best example is called as a multicellular animal. Multicellular animal. Multicells means the number of cells are present here. Can we count the stars in the sky? No way. Can we count the sand grains in the seashore? No way. Can we count the water drops in the sea? No way it is. At the same time, we can't count the cells present in the multicellular animals. Of course, like our, for example, human being. Now, way to count the cells, but approximate number we have to guess now. Okay. So, number of cells, mass of cells are present in the multicellular animal. So, most of the cells presenting in the internally and externally. So, for example, regularly we are washing our hands, we are taking bath regularly. By washing our hands, by taking bath regularly, number of cells automatically peeled off. These are all commonly called as the dead cells. Dead cells. In place of the dead cells, once again the body, of course, the epithelial cells presenting in our body can also form the new cells once again. By the process is called as mitosis once again. This credit only goes to for the mitosis. This process of mitosis not occurs on the surface of the body. Of course, in the internal parts of the body also. Either the digestive system, okay, or the reproductive system or respiratory system or circulatory system, any kind of one. Wherever it is necessary, whenever it is necessary. Wherever and whenever it is necessary, the cells number can gradually, continuously develop repeatedly, rapidly. Okay, depending upon the age conscious, the division process may automatically changes. Okay, during the growing stage, the mitosis process is gradually, of course, occurs very too rapidly. Number of cells are worn out in the internal parts of the body also now, whereas in the outer parts of the body now. Yeah. So, whereas both for internal and external parts of the body, the process of mitosis taken place. In place of that, new cells are forming now. That's why the first statement by the Rodolph Virchow, all the cells are forming from their pre-existing cells. The cells can give the chance to the next one now. Okay, so number of cells having this particular period. For example, human RBC have the life period of 120 days. Red blood cells. White blood cells also having nearly 12 to 13 days. Blood platelets also having nearly 10 to 12 days. Approximate days now. All the cells, all the cells having this particular time periods of their lifespan now. In place of this particular one, new cells are forming now. For example, when the lifespan of the red blood cells is completely clear, new red blood cells are forming from the bone marrow. When the cells of the skin is automatically terminated now, new cells are forming from the surface of epithelial cells now. Once again, the related tissues can also take the responsibility to form the new cells once again. Okay, this kind of continuous process occurs again and again, again and again to make the continuation of life of an organisms. I'm not saying about the human cells only having this kind of ability. All the animals and plants are having the same kind of an ability to produce, to regenerate again and again, to making the continuation of life once again. And clearly, what is meant by cell cycle? Clearly observe, cell cycle means we have to understand the process is called as mitosis. Mitosis means a kind of a fine thread-like structures are present in this one. Okay, this is a just imaginary diagram to you for our understanding easily now. So each and every cell division takes approximately nearly 40 to 60 minutes. Okay, this is the stage of the interphase. All the cells are in the resting mode, are in the resting period before the cell division. That is called as interphase period. Before cell division, when the cells are in the period of resting position, resting mode, then it is commonly called as an interperiod. I mean, that is called as interphase. Okay, in this particular mitosis, there are four phases are present: G phase, sorry, G1 phase, S phase, G2 phase, M phase. Clear it is. G1 stands for gap 1. Means here, before cell division process, within this particular period now, chromosome numbers or chromosomal replication ready to start. Division of chromosome chromatin material. You understand? This is the ending of the mitosis process. Okay, this is the ending of the mitosis process. After mitosis process is ending, G1 phase is ready to start now. Okay, chromosomal number, chromosomal replication starts now. Replication of chromosomal, duplication of chromosomal number is ready to start. In the first one is called as gap G1 gap 1 phase. Okay, so these chromosomes are ready to divide into two parts. For this purpose, the chromosome number is ready to replicate, ready to form, ready to synthesize. I mean, ready to form, ready to forming now. And here the S phase, S stands for synthesize. Chromosome number is ready to synthesize, making of chromosomes. Okay, a clear-cut division of making of chromosomes occurred in the part in the stage is called as S phase. This is the gap. This is the stage between the G1 and G2. Okay, next one is called as G2 phase, gap 2. Okay, this is the starting phase of the mitosis. Starting phase of the chromosomal replication. This is called as ending stage of chromosomal replication. Here now, end stage of chromosomal replication. Chromosomal replication is started here. Chromosomal synthesis making in the S phase, and chromosomal replication is ended. And this is the end stage of mitosis. This is the starting stage of mitosis here. G2 is the starting stage of mitosis and end stage of the chromosomal replication. G1 is the end stage of the mitosis and the starting stage of the chromosomal replication. Whereas S phase is the between gap. It is a stage between the gap 1 and gap 2. So S stands for here synthesis. Chromosomal making or synthesis is taken place within the part of the S stage now. Okay. After completion of this particular G2, when the chromosomal replication is completely over, then it is said to this particular one is called as M phase. Here the stage M is called as mitosis. Mitosis stage is started here. Means within the mitosis stage, once again, there are some more subphases are present: prophase, metaphase, anaphase, telophase. We will discuss later in the coming topic next week. No problem. It is no. So within the mitosis, these all stages are also completely occurred. All these stages are completely comes under as a M phase. To understanding this particular stage, Pothur Simha Rao, okay, also did a comprehensive works and clear-cut divisions of the HeLa cells. Of course, you can also see in the annexures in the textbook itself. No, clear it is no. So this is the cell cycle. Clear. This total particular periods taken nearly by 40 to 60 minutes itself. Now, first of all, listen. Cell division observed by Rudolf Virchow, Robert Remarque, and Walter Fleming, Wilhelm Rex, and again uh, George Gregor Mendel. And these particular stages, just now I told these stages first of all observed by Theodore Baveri. What stages I said, these sperm cells and for example egg cells and also forming, each and every matured cells can also form as the bone and muscles. These first complete stages are observed by the Theodore Baveri. So what is the stages of the mitosis? What are the steps presenting in the mitosis? We will discuss later in the coming topic. Okay. Thank you.